So, hi everyone. Welcome back to fifth Spatial Omics seminar series. And today we are excited to launch, and we would like to also have our first speaker from Europe. And currently we have two speakers, Paula and Marc, from Dr. Holger Haynes' team, and they're from Center for Genomic Regulation, Barcelona Institute of Science and Technology. And today we will talk about spatial transcriptomics and a tumor immune cell atlas, steps towards precision oncology. And you may have seen their work on Spotlight, and we were just chatting about that before we get started today. But we look forward to hear more details. And without further ado, I'd like to turn to Paula and Marc.
Okay, let me just confirm that we can see the full screen. Yep, okay. So thank you very much for inviting us, and we're very happy and excited to be able to present our work and our ongoing research. Um, so as you said, we are Paula Nieto and Marc Elosua from the CNAG-CRG, and we're in Holger Heinz team, the single cell genomics group. And today I want to talk to you about exactly this, the spatial transcriptomics and tumor immune-cell atlas, and the steps that this takes towards a more precision oncology. So very briefly, we're going to try to explain a little bit the introduction and motivation of Spotlight, a little bit how Spotlight works, some benchmarking results, and then Paula will come in to explain her work on the tumor immune-cell atlas and how it integrates with spatial technologies. So very briefly, because I'm sure we all know this slide. When we do bulk RNA-seq, we are grabbing a bunch of cells, we're sequencing all of them together, and we get an average profile expression of that tissue. When we do single cell RNA-seq, we go at this much finer grain resolution in which we are able to um, detect which reads or which genes each cell is expressing, and we get this much finer granularity and much finer understanding of the biology of the data set. But it has some limitations. Some of the most important limitations could be that you lose the spatial coordinates of these cells during the dissociation protocol, and that during this dissociation protocol, we may have some technical bias in the way that we capture certain cell types. Certain cell types will be easier or harder to collect with these data sets. And ultimately, with spatial transcriptomics, we are able to, or we aim to be able to profile biological information, in this case, transcriptome information, while maintaining its spatial location. And ideally, we would have what we see here. So we'd go from the fruit smoothie to the fruit bowl, and we would go to the fruit pie, in which we would see how all the cells or all the fruits lay out on top of the pie. But with most spatial transcriptomics technologies, what we have are these mini smoothies within on top of the tissue. And what we need is to we don't want to stop just at knowing which genes are being expressed within each one of these spots, but we want to get a finer understanding of which cell types are contributing those reads to each spot. And to do so, in the lab, we developed Spotlight, and this is a very brief graphical abstract. I'm going to walk through this quickly, and then I'm going to stop step by step. So with a single cell RNA-seq data set, we are able to train our model and obtain these cell type specific topic profiles. And then once we have this model trained, we can grab our spatial transcriptomics technology, obtain the spatial spots, run them through the model, and obtain capture location topic profiles. So once we have the cell type specific topic profiles and the capture location topic profiles, we just need to find which combination and at which proportion we need to fit each cell type to fit best the spots topic profile. And ultimately, we will obtain or we aim to obtain the predicted proportion of each cell type within each spot. So I think it's always nice to have a good understanding, or at least of an intuition of what's going on behind the tool and how it's working, just to be able to not just use it as a black box, but be able to really fully understand what's going on behind the curtains, and just be able to interpret the output much better. So before we start with the gene by cell matrix from our single cell reference dataset. And what we first do is we want to decompose this matrix into a gene by topic matrix and into a topic by cell matrix. And in this case, we assume that the number of topics is going to be equal to the number of cell types present in our single cell reference dataset. And therefore, what we do is that we assign each topic initially to one cell type. Therefore, we will initialize each column here, which is a topic, with the marker genes for one specific cell type. So for example, in the first column, we will initialize with genes belonging, or marker genes obtained, or differentially expressed genes for cell type, for the green cell type, and so on and so forth. And then for the coefficient matrix, we will assign a 1 or 0 depending on if the cell type belongs, or if that cell belongs to this cell type or not. So in this case, we will have something that looks like this in a toy example. The next step that we do is we perform this matrix factorization after this seeding of the initial matrices, and we obtain a gene by topics matrix. And in this matrix, what we have is that each column represents a topic, and what the model here aims to learn is the cell type, or the marker genes that are most important for that topic, which in turn is that cell type. And then on the other hand, we get which is the topic composition, or the topic, the topic composition for each cell. So once we have this topic composition for each cell, what we want is to condense this information, and we want to obtain cell type specific topic profiles. So we consolidate all of the topic information for all of the cells from the same cell type to obtain one cell type topic profile. And we can see here that the vast majority of cases, each cell type will be represented by one topic profile. This may not always be the case, since if we have not necessarily cell types, so if we have cell states transitioning between cell types, then we may have that one cell state is represented by 2 or 3 topics. Then the next step that the model does is it introduces the spatial transcriptomics data here, in which we have the gene by spot matrix. And we also pick back up the gene by topic matrix, which the gene by topic would be the basis that we use to map our gene or our count matrix to our topic matrix. And by running our spatial transcriptomics dataset through this basis, we're able to obtain these spot topic profiles. And we see here that they're much more mixed, meaning that there's a variety of cell types within these spots. And then ultimately, in the last step, we have these spots, which the composition is unknown, but we do know what their topic profile looks like. And we have these topic profiles that are cell type specific. And once we find the proper combination and the proper weight for each cell type, we are able to fit it to each spot's topic profile and obtain the proportion of each cell type within each spot. But how can we visualize this a little bit with our eyes and just get a sense of what's going on? First of all, we can visualize the coefficient matrix. So for example, we want to see if all the cells from the same cell type share the same topic profile. And for example, here we can see, for example, B cells, CD4 effector memory, CD4 naive memory, they all share the same topic profile distribution. So by looking at this plot, we can already see that the cell types that we identified are similar to each other, since once we run them through the model, we obtain a topic profile for all of these cells that is common. And then once we consolidate this information, we're able to obtain this topic by cell type and obtain this cell type specific topic profiles. And we can see here that all the cell types are very specific. The only one that appears to share a topic with another cell type would be SPP1 macrophages. But when we see which cell type it shares this or partially shares this topic profile is tumor-associated macrophages C1QC. So it makes sense that there would be some shared gene signature between these two cell types, since they're both macrophages. And then another thing that we might be interested in is looking at which are the marker genes that are driving these gene signatures, these topics, right? So in this case, we can do this. We can get this insight by looking at the basis matrix. And we can see that, for example, topic 1, if we go back, represents B cells. And when we see here, we can see that the top genes that the model has identified are classic canonical B cell markers, such as MS4A1 or CD79B. So we can have an understanding of which cell types are driving each topic, and thus which cell types are ultimately driving the deconvolution. So I think that selecting a good gene set to run this this process is very, very important. And at least myself, I always check after the deconvolution just to see and get a sense which are the genes that are driving each topic. So once we had the tool built, we wanted to test it on a real biological data. And the first thought that we had was to use it on a brain or to test it on brain, since it is a very known tissue, very well studied, but also very complex. So there's, on the one hand, there's a plethora of information, and we have multiple data sets, multiple resources where one can find information. And on the other hand, it's very complex, but it has very clear and marked structures within the tissue. So what we set out to do is to grab this data set from the Allen Institute, which is a single nuclei data set of the mouse brain, in which they profiled cortical regions and hippocampus. And it's contained by 45 cell types and over 76,000 cells. So we consider this a very complex data set, and we wanted to see if we were able to map this onto a vision data set. In this case would be a sagittal cut of a mouse brain put out by 10x, and see if we could recover these structures. So once we perform the decomposition of these spots with spotlight, we can see how we can clearly see the different layers of the brain. We can see of the cortical, or the different cortical layers. We can see how MICE2 cells are mapping the olfactory bulb. We can see how the hippocampus, the chronomonus, and the denta gyrus. I will show this in a little bit. But we're very happy that we were able to recover these structures right off the bat. This, I just wanted to show a little bit that we are able to map the different cortical layers of the mouse brain cortex. So brain L2-3 or layer L2-3, the outermost layer, then L4, L4-5IT a little bit further in, L5IT, L6 CT, and L6B. We can detect even this L6B, which is very, very fine structure. We were able to recover it, which it made us very happy. And then Some other examples of how we were able to recover these structures are, for example, the chronomonus 3, which would be this second section that we see here highlighted in black. We were able to map it exactly here and over here, which is also right there. The denta gyrus as well, over here and down here, as highlighted in black. And these are images obtained from the Allen Brain Institute. They do a great job at atlasing and putting out these public data sets that are invaluable to our research, and as well as chronomonus 2, that we see that it maps right over here, which is exactly where we detected, or exactly where it is when we image it. And then once we had seen that this works on a model organ, let's say, with known structures and known cell types, we wanted to see if we can validate it as well using a cancer data set. And to do this, we used this pancreatic ductal adenocarcinoma data set put out by Ruben Moncada and Itai Yanai. They put out this single-cell RNA-seq data set generated with DropSeq. And we can see here how there's 2 clonal cancer populations, one with different transcriptomic profiles, 4 different ductal populations, as well as immune cells and other cells present in the pancreas. And we mapped these onto the spatial transcriptomics data that they also provided in this paper. And I think of note here is that the spatial transcriptomics data is the original spatial transcriptomics data. So the spots in this case are 100 micrometers in diameter and spaced 200 micrometer by 200 micrometers. And it is very interesting to see how we have how we're able to map the cancer clones specifically to the cancer area here on the bottom right at much higher intensities. And we're able to map these central acinar ductal cells exclusively to the healthy region of the tissue. And then I think something that was very interesting is that we also had these, or the data set also contained these hypoxic-like ductal cells, which are healthy ductal cells but are expressing hypoxia gene programs. And we see how in this case, these healthy cells map exclusively to the healthy part of the tissue, but here they map at a much higher intensity within the tumoral or cancerous compartment of the tissue, indicating that even though there are tumor cells within that region, there are still healthy cells within that compartment that have had to adapt their gene programs to survive in this hypoxic environment generated by the tumor. So we're very happy with these results. And the last thing that we wanted to set out to do was to benchmark our tool to other deconvolution tools, bulk deconvolution tools existing at the moment, as well as RCTD. And we see how Spotlight, and I wanted to assess this in 2 ways. First, we wanted to assess it in a way to see if we could detect if a cell type was present or absent in a spot. And we did, and here on the plot on the left. And also we wanted to see if the proportion at which we mapped each cell type to each synthetic mixture that we generated, we were able to recover it. And we can see how Spotlight was able to perform very well with high accuracy and a high F1 score. And then it was able also to perform very well at predicting the proportion of the cell types, although RCTD in this case and music were also very, very good performers. So moving on to another application or a more biological application is the Tumor Immune-Cell Atlas that Paula is working on in the lab. And I'm gonna allow her to continue the presentation.
Thank you, Marc. Yeah, so we developed this Tumor Immune-Cell Atlas in I will tell you after how we use it with Spotlight. But basically, we took a lot of samples from the publicly available data sets from the literature, single cell data sets, and we integrated them to study the immune cells. So we incorporated into the atlas 13 cancer types, from over 200 patients and more than 500,000 cells. And yeah, this gives us a broad spectrum of cells that we can study in this tumor immune microenvironment. And yeah, you can see on the left the different cancer types that we have in the atlas, so the number of cells for each of them and number of patients. The first is lung cancer, melanoma, colorectal, breast, etc. We have different cancer types, different numbers of cells, and different numbers of patients, which gives us more range of cells and more variability to study this. And we use integration tools to put this all together. And because this, as I mentioned, came from different data sets, also from different single cell technologies. So it was difficult to have it all integrated, but I think also because we have a lot of variability, we were able to conserve that variability, but also find the commonalities because we only are studying the immune cells and not the cancer cells. And yeah, here you can see we're showing here the 25 cell types that we found. We then reclustered a bit further to find some minor populations, some inside the B cells, etc. But the ones that we're showing here is the main 25 clusters. We have a lot of resolution, for example, in the T cell compartment and the macrophage compartment, and you can see in this violin plot that we were able to use canonical markers to annotate these data sets, so T cell markers, B cell markers, and so on. Yeah, also you can see here in the plot on the right that for the different cell types that we annotated, basically all of them are represented by a high number of patients, but we also recovered some minor or less abundant cell types that were only present in some patients. And I think this is the advantage of looking at this from an atlas perspective or from an integrated perspective, that we are able again to capture commonalities, but also to see minor differences. And yeah, this is also what we wanted to explore a bit further, and I won't go into many details here. But we use this many patients that we have and these many cell types we were able to annotate to develop a patient stratification method. So it's basically a clustering algorithm that groups the patients according to their immune profile. So the immune cells that are in their tumor microenvironment. And you see at the bottom of the slide that we have 6 clusters, and then we have different cancer types in the clusters. But yeah, basically this represents again these differences in the patients, but and differences in the cancer types, but commonalities across them. But yeah, if you want later, we can discuss a bit more about this. But yeah, this is one of the applications that we use for the atlas, and the other one was to automatically annotate cell types. So if you have worked with single cell data, you know that we often spend a lot of time annotating the cells, right? So we wanted to also create this tool so that we could annotate external data sets using our atlas. And this is what we tested. So for this, we generated in-house samples. We sequenced them for human and for mouse. So for the human, we have a liver metastasis from uveal melanoma patient, and in the case of the mouse, we have a colorectal cancer sample. And you can see by the colors are the ones that the same that we use for the atlas. So we were able to project these new samples, provided that they are immune cells, to our reference atlas, and we were able to annotate them. But another way that you could do this, and that you see on the right heat map, is that other than projecting them, you can also use or compare the marker genes. So we were also able to annotate these different clusters or these different cells by comparing the the clusters, or sorry, the markers from our clusters in our query datasets to the markers from the atlas. And then the final application that we use the atlas for is to for the spatial deconvolution in combination with Spotlight. And I think this is where it gets to the most you know, the most value of both the atlas and also bypassing the limitations of this sequencing technology. So I will present here an example that we did, which is oropharyngeal carcinoma, and you can see the slide on the left, and on the right you can see you know, basic clustering and supervised clustering that we did for the spots. So this is the same technology that Marc mentioned in the beginning. So each of the spots is a mixture of cells. So far we have 4 regions, 2 carcinoma regions, 1 stromal region, and 1 that we annotated it or identified it as immune cell rich, which is this purple one. And what we did is we used the atlas that I presented as a reference to deconvolute this spot. So we were able to again map these different immune cell types onto this slide. And you can see, for example, in the immune-rich region that we were seeing in the beginning, that is very high in B cells. So I don't know if you can see it very well because we have this pie chart plots for each of the spots, but this one is enriched in B cells. For the other, for example, carcinoma regions, we see that this is more enriched in T cells, like effector memory T cells or T helper cells. And this is very nice because often this can help also diagnose or understand how this cancer is behaving, right? So is it infiltrated, or is it preventing the immune cells to enter the cancer? So this is something very nice that we can do pretty fast by having a reference and Spotlight together. Yeah, and here I'm showing the same thing, but in this case, instead of the pie chart plots, we see the abundance of these different cell types in each of the spots. Okay, so in the first one, in the B cells, we see that it's mainly and very highly expressed in this immune-rich region that we were able to identify, while as, for example, the plasma B cells are more like on the outer layer of this section. We can also see macrophages, the SPP1 macrophages are basically spread throughout the tissue, or mainly in the cancer regions, but are basically spread. And for example, these other tumor-associated macrophages is cluster is a bit outside, you know, this region. So it would be complementary to these other macrophages. And if we look at the proliferation, for example, in this case, this is taking into account not only the immune cells, but also the cancer cells that are proliferating. We see very nice how it matches some of the clusters or some of the regions that we were able to identify before in these spots that we have. And another thing that we can see with this is we can both compare or find regions of cells that are colocalizing, so we can find cells that are expressing in the same regions, but we can also see with markers like canonical markers that we can really find these cells in these regions. So on the left, you see the cytotoxic CD8 T cells and the T regulatory T cells, sorry, and we can see that that it seems like they're colocalizing in the slide, and we can also contrast this by looking at Treg canonical markers such as FOXP3 or CTLA4. That the expression of these genes matches the predicted location of the Tregs, and the same thing for cytotoxic markers such as DNLY or NKG7. We see that it matches the predicted presence of cytotoxic by Spotlight. And other things that we can look at is if we are interested in understanding these 4 regions that we were able to annotate on our slide, we can compare the composition of different cell types. For example, here you can see B cells, macrophages, and tumor-associated macrophages, and we see the B cells, as we were seeing before, are very enriched in this purple region, which is the immune cell rich region in the slide. While as, for example, the macrophages, STP1, that we were also seeing before, are in the carcinoma region, so in regions 1 and 2, which are the green and the blue ones in the slide on the right. And for example, the other macrophages, the tumor-associated macrophages, are mainly in the stromal regions. So this is very nice with what we can see and all the information that we can get for the cells that are co-localizing and how these different regions are composed. And we also, another thing that we did is we studied the correlation of these cell types by look, you can see this in the heat map, the correlation of the cell types, and what we were able to see with these correlation is that we have differentiated regions. So you can see the green region would be the STP1 high region, which is the carcinoma region. And we see how like these cell types co-localize in this region. Whereas, for example, the pinkish region, the stromal region, is very high in other cell types such as T1QC TAMs or Tregs or CD8 T cells, etc. And this purple region, the B cell high region, which is the immune cell rich region, also is a separate group. So this is we can really get specific to which cells are co-localizing and where, and this basically demonstrates that what we were seeing with the deconvolution is really something that we can really map back to the tissue and really understand how this tumor is behaving. And yeah, I think that's it, at least for now. We wouldn't like to finish without thanking the organizers for allowing us to present in this space, and also all the team members in our lab that helped make this project possible. So of course Holger for his leading in this project, his leading role in this project, and also all the lab members that process the samples that we map back to the atlas, or Juan that helped with the annotation of the tissues, and basically everyone that helped us, and without whose work this project wouldn't have been possible. And of course, thank you all for listening to us. And yeah, finally, if you are interested in using Spotlight, Marc has created a very interesting and thorough tutorial in the GitHub, and you can use his tool right now, and also check the paper. And if you're interested in the atlas, it's the dataset is public, so you can find it. And yeah, we also have a repository with all the code that we use to generate it in a preprint. Thank you.
Okay, it's a terrific talk. Thank you both. Thank you very much. So I think whoever has question, you can unmute yourself and ask. You can also use in the chat box, but I think we can be relatively informal. That's the format we try to kind of keep for this, rather than just a webinar. We decided not to do just a webinar. Instead, we really want to have this kind of very interactive format here.
Mm-hmm.
Yeah, I think when people are still thinking about their questions, I do have quite a few. I'm probably gonna ask the first question. So that's the question I actually asked my student this morning. So when you do your spotlight decomposition, you have kind of percentage of different cell types within each pixel, each spot. So how did you define that percentage? So what's the kind of criteria?
Right. So if we go back to here to identify the proportion of each cell type,
Mm-hmm.
right? So once we see that the spot's topic profile looks like this, the idea would be to find the percentage of each cell type that would contribute to this spot. So for example, in this one, an idea could be that this cell type contributes 30%, this one contributes 20%, this one contributes 20%, and the pink cell type in turn would contribute another 15%, for example. So we just try to, by using non-negative least squares, we try to find which cell types and at which proportion do we need their topic or their representative topic profile to resemble the most the the spot topic profile that we're trying to fit.
Mm. So basically, each cell type is a transcriptional profile, right? So with
Exactly.
a finite set of genes.
Mhm.
So I'm just thinking about what the math here. So you kind of set a different weight factors for different cell types, and then you do kind of fitting and see which what weight factors for each cell type can give you the best fit.
Exactly.
Okay, okay. I
Exactly.
think my question too is, so I kind of noticed in your data that the brain atlas, the brain spatial omics sequencing data, that really surprised me. Seems like most spots are kind of dominated by just one cell type, but those spots are very big, right? So it's unlike they are just one cell or 1.5 cell, but it's very striking. I think most of them are just like 80%, like a 70% or even 100% just one cell. So that's, yeah, maybe you can explain first.
So I by no means I consider myself a brain expert or a neurologist. So
maybe I
take
can
it with.
try to explain it a little bit more, because I had similar kind of things also with my data that happens exactly the same. And we actually both found out that it is due to the underlying data, like the data that you basically integrate for Spotlight. Because in this data, what you see here, it's mainly layer structures and bigger regions, but if you also insert smaller sub-clusters of cell types, you get also different pie charts, basically.
Okay, so you think that's the selection of your data set to do spotlight decomposition
of the reference data set. Exactly.
Okay. Okay.
So if there were, for example, in this data set, we only detect astrocytes in the outermost layer, but we would expect to find astrocytes intermixing with the neurons as well.
Okay.
So if the reference single cell RNA-seq data set had these different subpopulations of astrocytes, we would expect them to map more partially to these spots.
Okay, I got it. But I thought there could be another reason for biological. So within a certain region of the tissue, right? So the cells and their neighbors actually are quite similar. Maybe in your case, you just define that as one type of cell, even though you have two cells there, but they pretty much look like each other, right? Yeah, could that be a?
I think for the brain, I think they did a very thorough selection of these cell types. So I think they even sometimes mark them fluorescently to be able to select some cell types that are specific. So I think that I really trust the cell type annotation that they did. And as I said, the brain has this very defined structures where we find mostly one cell type in each structure, even though there may be this other adjuvant cell types. But for example, when we show in the oropharyngeal slides, there is a lot more mixing of cell types within each spot. There's the pie charts are a lot less monolithic in the sense that they only contain one cell type. They contain much more diversity.
Okay.
Actually, kind of wondering also maybe that it also could be due to dropout events on the spots, basically.
Maybe. I think that dropout is something that if we work with the single cell or droplet-based sequencing technologies, is that something that we need to live with?
Yeah.
Yeah, I think Caleb asked a question in a Dropbox, in a chat box. If you, I can read for you, Marc.
Yes,
That's in addition to the percentage of each cell type, either a way to get the number of cells in each spot, so you can use the density of cells for region identification.
So this is something that we're talking just before, I think, with Joy. And this is something that Spotlight doesn't currently do at the moment, but it's definitely something that we're thinking of implementing. And we believe that it would add a great deal of information. Yes.
How do you think about kind of if you want to integrate such a thing, and kind of how do you want to do that? Because I've thought about it as well, kind of like how to construct basically that. Because the only thing that I came up is by using basically housekeeper genes, Marcus.
To try to identify how many cells fall within each spot, more or less.
Yes.
I think you will need to go with more AI or machine learning algorithms that you could, how's it called, section the tissue and then identify or just look at the tissue, identify where the nuclei or the cell walls are, and then try to see how many of those fall within the specific coordinates of each spot.
But how do you correct them for the RNA hybridization kind of fuzzy measure, basically?
I think there are a lot of interesting questions here, and I'm sure there's a lot of approaches, but I think I would try, or at least the approach that first comes to mind is I would try to, with algorithms that allow you to do the segmentation of the tissue and identify the cell types, and then once you have these cell types identified, see how many of these fall within each spot.
Yeah, I would say just do better image integration, so you
higher resolution.
Yeah, you can actually see how many cells in each spot rather than just a computational guess, right? Yeah. All right, so any more questions? I think Ahmet, you always ask a bunch of questions.
Sure. So I was curious about the assembly of the data for the second part, the Immune Atlas in tumors. So here, when you assemble data from literature, how do you standardize this, right? Because you profile these immune cells, and in the next slide, I think you showed a really nice map of these, and then the next one. So how do you standardize these things? Because biology is different, right? It could be from primary tissue, a different part of the tissue, or it could be, a different stage. Do you take into account these biological differences?
Yeah, so definitely this is a challenge. And yeah, so first of all, the first layer of heterogeneity, I think, was removed when we only studied the immune cells. So we only considered tumor samples, and from that only the immune cells, because the tumors are actually much more heterogeneous even between patients, but the immune cells are much more conserved. And also something that was affecting a lot the variability in our data set was the sequencing technology. So in this, we have a mix of 10x sequencing. We also had MAR6, SMART6 2. So I think that was like the big source of variability. And in fact, we benchmarked different tools or different approaches of the integration. So in this case, we went with Seurat, which was the one that was performing the best. But there are some papers that say, you know, Harmony, for example, is better at biological preservation. But for example, in our case, because, you know, we had these variable sources, I mean, not sources, sequencing technologies, Harmony was not performing that good at conserving the variability. So this is definitely a trade-off, you know, between having good resolution, having good integration, and wanting, you know, the most refined differences. But in our case, Seurat version 3 worked best, but I think that so far, even new technologies, new algorithms and packages have come out that would definitely be worth trying.
I see. Yeah, I think the immune cell, you know, it's a very complex map, right? And it's kind of changing at different time points. So actually there's a new grant called the Tissue Time Machine, right? It's almost like a time machine. The immune cells actually go at different times to different locations. So that's actually an interesting one. I think this is interesting for the second part. And for the first part, Marc, the spots are really nice and they're passing different anatomical regions. But I'm curious if you can do some kind of like a sliding window approach to increase resolution of spot size, right? Spot size is fixed, but can you do some kind of a sliding window and learn the sub-spot resolution from this?
The sunspot or the interspot?
Interspot, yeah. For instance, you're sliding your predictions, then it's almost like looking at the derivative of the signal, right? Not the signal by itself, right? And they will get more sharp. So then you can actually, so there are actually microscopy techniques using these sliding window approach to increase resolution. I'm not sure if that can be improved in molecular profiling of this.
I actually, this is a super interesting idea, and I was reading a paper a couple of weeks ago that they actually do this. I think it's called base space. That they do this, try to build up the space or like try to give a sense of what is going on, not in a way to decompose the spots, but try to spread the transcript information. So try to mix a little bit, or try to identify what's going on in between these regions in a way that a little bit what you were saying. So if four spots share a certain gene program, for example, then most likely in between those spots, that gene program will also be present, right?
Okay. Yeah, exactly. I think that will be pretty cool to digitally increase the information, right? By looking at the space and assume it's continuum, then discretize it, and go back and forth between different discrete steps. And
definitely, yeah.
Exciting. Any other questions from our audience? So it's 2:54. So if not, maybe we could thank our speakers today, Paula and Marc. Thank you so much. And I think we have this clapping sign that we can use. I need to find out. Okay, here. Yeah. Thank you so much for sharing your interesting and hard work that you did in Spain. And so next week, we will continue with an industry speaker from Rebus Biosystems, and Erin Colleen Davis will be presenting the talk, and it will be about the system that they developed, Rebus ESPER, and it's a quantitative single molecule, single cell spatial data with subcellular resolution. I think we'd like to hear this talk with more details because it's a very new system in the market. And with this, thank you guys so much for participating today. And we look forward to seeing you guys next week.
Thank you very much for having us and for giving us this opportunity. I put the name of the base space package in the chat in case you want to take a look. But I think it's a very cool tool available.
Yeah, we'll
Yeah.
take a look. Thank you so much.
Mhm.
Thank you. Have a good day, everyone.
Thank
Have
you
a
very
good
much,
day.
and have a good
Bye.
weekend.
Have a good
Yeah.
You
weekend.
too.
Thank you.
Bye.
Bye-bye.